In this video, we'll talk about general mechanisms that enzymes use to catalyze reactions. Here are our objectives. The first is to describe the basic mechanistic principles used by enzymes to increase reaction rates. We'll talk about the role that most vitamins, especially the B vitamins, and many minerals play in providing chemical capabilities at enzyme active sites. We'll talk about why the pH of blood in most tissues must be kept within such a narrow range. In addition, of course, to keeping the rate of oxygen delivery appropriate. Remember that hemoglobin and the Bohr effect is very pH dependent. And lastly, we'll talk about why a fever or hyperthermia results in increased metabolic rate. So the human genome encodes thousands of different enzymes, and metabolic enzymes are a fraction of this. And I would just want to remind you, I'm showing you here our metabolic map, because every one of these arrows on the map is a separate enzyme, right? These are all reactions, chemical reactions catalyzed by different enzymes. So let's start off talking about an enzyme in the pyrimidine biosynthetic pathway. This is in the purple region of the metabolic map. And the reaction we'll talk about is here, right in the middle of the pathway. And it's a fairly simple reaction of removing a carbon dioxide from auristatine 5' monophosphate, or OMP, to make the pyrimidine UMP. So this is the basics of the pathway in pyrimidine biosynthesis, where I won't go through all of this, but the reaction we're interested in is this one down here. It's the sixth reaction, OMP decarboxylase. So again, we're losing a carbon dioxide group from here, from OMP. And so again, this is the reaction. We're looking at down below at a ribbon diagram of the backbone of part of the enzyme, and we're peering into the enzyme active site. Here's a substrate analog that's chemically stable, so it could be crystallized in this enzyme, so we could determine the structure of it. And then what's shown in the cyan color are amino acid side chains that are very carefully and appropriately aligned, that the enzymes evolved for them to be very appropriately aligned and positioned with this substrate to allow the reaction to occur. Okay, so here we are right in the active site. And again, this is a fairly simple reaction. Here is our substrate at the enzyme active site. We're not going to go into the mechanism and details of this, but I just want you to realize we have an aspartic acid side chain, right? So a negatively charged side chain, a positively charged lysine side chain. These are very important. The exact positioning of them is very important. And in order for this reaction to occur, we really need the acid and the base there and appropriately positioned. Okay. So this reaction, quite a simple reaction. An incredible thing about it, however, is if there is no enzyme, this reaction takes more than 78 million years to occur. This is an incredibly difficult reaction. There's a very high free energy barrier to the removal of a carbon dioxide at neutral pH and at 37 degrees body temperature. This carbon dioxide just doesn't come up. It's incredibly slow. And yet the half-life at the enzyme active site is less than 0.02 seconds. So this enzyme is greatly increasing the rate of this reaction. In fact, it has a rate enhancement of 140 quadrillion fold. Okay, so here's a second enzyme example. This is of delta ALA synthase, and we've talked about this previously. This catalyzes the first step in heme biosynthesis. Okay, it's the enzyme that takes succinyl-CoA and glycine, puts them together, releases coenzyme A and a carbon dioxide to make delta aminolevulinic acid, or ALA. And here it is on the metabolic map. Here's the structure. It's a homodimer. Okay, again, we're looking at a ribbon diagram that's just showing the backbone of the enzyme. But if we look into the active site, it's got a fairly large active site. Here's succinyl-CoA. Most of this is the coenzyme A, which is very large. And we see a glycine, and there's glycine here. And pyridoxal phosphate is a vitamin B6 derivative that's required at the enzyme active site. Okay, so here's another view of that same active site, and we're looking down in it like a pocket, and it's shown in blue around as a modeling of what the enzyme is. So here's coenzyme A in bright green attached to succinate. We have glycine, the little amino acid glycine, and here it's shown attached to the vitamin B6 derivative pyridoxal phosphate, or PLP. As shown here, this is the enzyme that's mutated in X-linked sideroblastic anemia in people. All right, and if we look at detail at the different amino acid side chains around this enzyme active site, and again, we're not going to really look at these one by one in detail, but I just want to show you that there's lots of different kinds of interactions as shown by these green semicircles here, hydrophobic interactions, and here we have hydrogen bonding interactions, all very specific for different parts of the substrates. So lots and lots of amino acids, lots of different binding interactions. These amino acids that are highlighted in red here are amino acids that are mutated to cause X-linked sideroblastic anemia. Okay, and we know that this disease, it's a genetic disease. It can often be cured by high doses of vitamin B6, pharmacologic doses of this vitamin. And the question for you is, why do you think this works? Okay, so if we, let me go back for a second. So what we think, the reason it works is that if there's very high levels of vitamin B6, this the vitamin derivative can actually help stabilize the whole enzyme structure, stabilize this enzyme active site, and allow the enzyme to work well enough to prevent this type of anemia. Okay, so how does this enzyme work? Again, we're not going to go into the details of it all. It's a pretty complicated mechanism. But what I do want you to recognize is here's a positively charged histidine amino acid. This is very pH sensitive. It's protonation. Here we have a lysine side chain that's covalently attached to the vitamin B6 derivative. This is pyridoxal phosphate structure here. The glycine comes in, gets covalently attached to that vitamin derivative. Then we have attack of the lysine side chain, a bunch of arrow pushing here. Okay, again, pulling at the importance of these lysine side chains involved. And then the rest of the reaction occurs and we have ALA synthesized. Feel free to look at the details of this if you're interested. Okay, but what I want to point out is sort of to find the commonalities here, the big picture take-home messages, right? So the substrates, they bind to the enzyme active sites, right? So these are the molecules that are going to be changed from the substrate form to the product form, okay? And the active site is where these chemical transformations occur. The active site amino acids of an enzyme often act as acids and bases in the reaction, and the enzyme activity is therefore often very pH sensitive, right? If we think about the thousands of different enzymes in our bodies, each one has a very specific need for the pH of its amino acid side chains for the reactions to work at the right speed. And this is ultimately why we have to maintain our body and our blood's pH to really very tightly around pH 7.4. Okay, the specificity of the reaction, right? So an enzyme usually only works with a single substrate or maybe a limited number of substrates that are quite similar to each other. And that's dictated by that exact amino acid structure of the enzyme active site. And many reactions, just like the one I showed for delta ALA synthase, require chemical capabilities that are not available just from the 20 amino acids. So these enzymes need cofactors or coenzymes that are often made from the vitamins we get from our diets or the minerals we get from our diets, or both of these, in order for the enzyme to work. So you may have noticed on these images I've showed you, the active site pockets of enzymes are often only a very small part of the entire enzyme structure. And the question for you is, why do you think that might be? So we think there are several answers here. One is that in order to get the very exquisitely sensitive structure of that enzyme active site, you need the rest of the protein to get those amino acids in just the right places. There's also parts of enzymes, parts of the proteins that are required for the regulation. And we'll talk about that in a separate video, different ways that enzymes can be regulated. And there can be, binding of other small molecules to increase or decrease the activity, binding of other proteins, etc. Okay, so those are two of the reasons. All right, so we've talked about some specific ways that enzymes increase rates of reaction. But I want to finish by just talking about sort of one general theory of enzyme catalysis. And that involves thinking about how enzymes lower the free energy of the transition state of the reactions, okay, that they catalyze. So let me just kind of slowly step through this. So enzymes alter the kinetics or the rates of reaction, right? They're catalysts. That's the definition of a catalyst. But they don't change the thermodynamics, which means they don't change the equilibrium between the substrates and products of a reaction. So let's go back to that first enzyme we started with, OMP decarboxylase. Here's the reaction, right? We know the non-catalyzed reaction is very slow. So the rate constant for this is 2.8 times 10 to the minus 16 per second, right? So this takes millions of years to occur. It's very slow. And the reason is because so if we're looking here at a free energy level diagram, right? We have the free energy of the substrate and the product, okay? So thermodynamically, this is energetically a favorable reaction. The product has a lower free energy than the substrate, but the reaction is so very slow because the transition state, the amount of energy that OMP must get in order to release its carbon dioxide is enormous. There's a very high free energy barrier to reach the transition state for this reaction. And because of that very high barrier of energy, the uncatalyzed reaction is incredibly slow. So what happens when we add the enzyme? Okay, so here, what's called the K-cat for this enzyme, or the turnover number, is much, much faster. So one molecule of this enzyme saturated with plenty of OMP provided can create 39 molecules of UMP in every second. It turns over the reaction very quickly. Okay, so in milliseconds, it's creating product. Right? So how does it do it? Well, at first it binds at the enzyme active site. So the substrate binds at the enzyme active site. That's what I'm showing here. We have an intermediate now of a bound form. And the free energy of the transition state is brought greatly down by the binding of the enzyme to that substrate. So where we had this enormously high transition state and a very slow reaction, bound at the enzyme active site, that transition state free energy is much, much lower, allowing this reaction to go much more quickly. But you'll notice that the free energy of OMP compared to UMP has not changed. The enzyme does not change that. That's why we say that enzymes alter the kinetics, the rates, but not the thermodynamics of a reaction. Okay. So the other enzyme we looked at, delta ALA synthase, this image shows nicely the interactions at that active site. There's a huge number of interactions involved in lowering the free energy barrier to the reaction, right? And let's look at another one that's sort of a nice, simple model to look at here. And that is, we've talked previously about in the heme biosynthetic pathway, ferrochelatase, right, which puts the iron in the middle of the protoporphyrin to make heme. All right, so that's shown here. This is the reaction catalyzed by ferrochelatase. And it was predicted by looking at this reaction that what had to happen was that this flat heme, the protoporphyrin molecule, had to be puckered, had to be sort of bent outward so that the iron could fit in the middle, because otherwise it couldn't just fit in by itself. And what's shown in yellow here is the proposed structure of what that looks like, this puckering. And a very close analysis of the enzyme active site showed that it evolved to perfectly fit a puckered porphyrin-like molecule, and that binding interactions between the protoporphyrin and the enzyme allowed the protoporphyrin to make this puckered structure and iron to quickly and easily bind it, right? And in fact, one test of this idea was making a stable molecule that looks like this, that's puckered. And they actually did this by putting a methyl group right in the center, which causes the protoporphyrin to pucker like that. And it binds incredibly tightly to the enzyme. And that's just good evidence that the enzyme active site really did evolve to bind tightly to such a puckered molecule. So let's sort of summarize what we have so far. Enzymes increase rates of reaction under physiologic conditions. So this means, if you remember back to organic chemistry lab, when you were trying to make reaction rates go faster, you could add strong bases or strong acids or boil the reaction in order to make them occur. Well, those things can't happen in the body. We have to have enzyme or reactions that occur really fast in microseconds or milliseconds, but they have to occur under physiologic conditions. And enzyme active sites allow us to do this. Enzymes are regenerated after each cycle. Okay, so they're catalysts, they're not used up. They can do one reaction, then quickly another reaction, quickly another reaction, and they don't change the equilibrium of the reaction. The reactions are occurring at what we call active sites. That's the part of the enzyme that binds the substrates and converts them to products. And the active site structure, often plus cofactors, many of which are vitamin derived, has evolved to lower the free energy of the transition state of the reaction. Okay, so one last thing. Let's put this a little bit of a clinical context. We have a 1-year-old neonate is found to have a fever, 39 degrees C, so about a 2 degrees Celsius fever. He's at elevated risk for developing hypoglycemia, so he should be watched very carefully to see if he develops hypoglycemia. So why is this? Why would a fever lead to something like hypoglycemia? Well, this all gets back to the fact that chemical reactions and enzyme catalyzed reactions, the rate of them increases with increased temperature, okay? About a 12% increase in reaction rate for every degree Celsius, right? So with this neonate who has a 2-degree fever, that means that his metabolic rate is about 24% higher than normal. Right? And that is not taking into account why he has the fever. There might be other things going on. And infants with their big brains, their big heads, actually use a lot of glucose. So hypoglycemia becomes a real issue here. And it all has to do with the fact that all the enzyme catalyzed reactions are going that much faster with the increased in temperature. Okay, so this gets us back to our objectives, which I think we covered each of these, ending with why we have a fever with increased, why a fever causes increased metabolic rate. All right, well, thanks for participating.